Welcome to the 7th edition of the Liver Seminar. And it is for me a great pleasure to introduce Dr. Robert Schwabe. Robert is an outstanding international researcher, tenure-track professor, and director of the Digestive and Liver Disease Research Center at Columbia University. He has several active fundings from the NIH, and he has mentored several students and international fellows, including myself. During the last 20 years, Robert has dedicated to investigate the mechanisms that promote the progression of chronic liver diseases and the biology of liver cancer, with a main focus on the role of hepatic stellate cells, as reflected by his outstanding last publication in JCI, Cancer Cells, and Nature. So please, Robert.
Well, I'm very happy to be here. I'm also very happy to see Sylvia here. I have to say, I mean, she was a long time in my lab, and I'm very proud to see that she now has her own lab. She has her own funding and is really developing well. So it's really a great pleasure to be here and see these things happening. So I'm going to talk about some work on stellate cells today. I called it Stellate Cells, Dr. Jekyll and Mr. Hyde, because I want to highlight a little bit unknown and also opposing function of stellate cells. So they're kind of the good guys and the bad guys at the same time. And a lot of this data is going to be unpublished. So I don't have any conflict of interest for this presentation, but I have a kind of a scientific conflict because I didn't quite know what to choose, because I know that a lot of you guys are also interested in hepatocellular carcinoma or other types of liver cancer. So my lab is actually doing a lot of work on stellate cells, but we're really focusing quite a bit now also on liver cancer therapy, because I feel like there's a lot of work now on pathogenesis and understanding how things are going, but there's very little work on focusing on improving therapies. So we've developed now systems together with other people where we can knock out any gene or activate genes via CRISPR-I and CRISPR-A. We can do this within weeks. It's amazing. The models don't take so long. And the gap we want to kind of address is the, how does this, just trying to, this works. So there are no highly efficient therapies for tumor cells in HCC. We have the tumor microenvironment focused therapies that work, fairly well, but overall, patients still on average die after 18 months or so, even with these very good therapies. So we can do a lot better. So our idea is to find tumor cell targeted therapies that can synergize with immune checkpoint inhibitors and anti-VEGF. So we have triple synergy, maybe even quadruple synergy. But I'm not going to talk about this today, because I talk about stellate cells. But if anybody's interested, they can ask me. So I want to start a little bit with an introduction on stellate cells. They were discovered by Carl von Kupffer, so actually they should be called Kupffer cells. I don't know who mixed this up, but at one point there was a lot of forgetting about stellate cells, rediscovering. So they were also called Ito cells because in the 1950s, a Japanese researcher named Ito found them by electron microscopy. He called them fat storing cells, and he already kind of showed the association with collagen fibers. And then in the 1980s, people started to isolate them. Scott Friedman was the first one. He didn't kind of invent a method to isolate stellate cells, but he used it to show that stellate cells make the most collagen among all cells in the liver. And since then, stellate cell research has exploded. Until then, it was kind of, I think these cells were a little bit ignored. I think you guys are kind of the experts. That's why I'm going to go really quick. Why is this all important? So a lot of people still die from chronic liver disease, and the main causes of death are cirrhosis and its complications like liver failure, bleeding, but also the development of liver cancer. And there obviously any type of chronic disease can lead to this. There are four main causes. But for the viral hepatitis, especially hepatitis C, but also with vaccination for B, these are becoming less relevant. And the alcohol is obviously remaining a problem, but now the big thing is the fatty liver called MASLD and MASH nowadays. I'm not going to try to pronounce the whole thing. It would take me half my talk. And you guys also know that the incidence is really high. This is showing the US incidence, but the global incidence is close, for fatty liver, is close to 40% now in adults. I mean, that's crazy. So I'm going to talk about stellate cells. They, as you know, come in two flavors, quiescent stellate cells and the activated ones, which are well described. I mean, they have been the focus of the field. They make the collagen. They're the bad guys. And I think we've learned a lot about them. But we don't really know much about quiescent stellate cells. I mean, you have to think about these cells can only do bad things. They must be in the liver to, serve some purpose. And we know that they store vitamin A, but is that all they do? Probably not. So, as mentioned, most people in the fibroblast field, in other organs and cancer, also see them as bad guys. But we think they have some protective or restorative functions, and that really it's the balance between these good and bad functions that also during disease progression gets lost. But that we always should keep in mind that there are probably some good functions, and we don't only want to get rid of the bad functions, we want to kind of restore the good functions. That's kind of the hypothesis. Yeah. So going to talk about two parts. So the first part is really these novel data on stellate cells in the normal and early injured liver. And the second part is published data on HCC. Second part is a little bit shorter. So in this next slide I'm showing you is also published. I mean, we already know that stellate cells are a main source for myofibroblasts. So this is a paper we published in 2013, where we kind of traced the stellate cells using the ALRAD-Cre we generated together with another transgene, the collagen GFP. And we showed that pretty much all of these collagen producing cells in a mouse are also labeled by ALRAD-Cre, which in our interpretation means they're stellate cell derived. It's not a 100% clean system. It's not inducible. So, you could have other cells expressing ALRAD, or during development things could change, but, we think it's pretty good. And we've done this in multiple models. Here you have the toxic models on the right. And then there were the biliary models like a BDL, but also other models. And you see in all these, on average, it's like 85 to 95% of the collagen producing cells were labeled by the ALRAD-Cre, meaning in our interpretation, they're stellate cell derived. So this is in mice. It's all fine. We all like to do mouse studies, but they're not 100% relevant. We need to look at the patients. So basically, in the second part, I'm going to talk about, again, the two functions of stellate cells, disease-promoting and protective, but in the context of cancer. And that's published data, but it still highlights the kind of diverse functions of stellate cells. I think I can almost skip this introduction. You know that a lot of people die, that liver cancer is increasing as opposed to many other solid cancers, and that there's a lot of fibrosis in these tumors. So we can start with the data. So we wanted to know what stellate cells do in liver cancer. Obviously, there's a strong link between fibrosis and cancer in HCC and cholangiocarcinoma. So I have one slide here showing you all the stuff we did with stellate cells to understand their role in cancer. So we mainly, but not exclusively, used again the ALRAD-Cre mice, and we activated, inactivated, and depleted stellate cells. And I think I'm just going to show you the kind of principal ideas, not all the detailed data. So first, we activate stellate cells by knocking out a repressor called LHX2. When we do that, and it's kind of been published before that LHX2 is a repressor. When you knock it out, you get more fibrosis, as we see, and we have more fibrosis. Here, a DN model of cancer, we get more cancer growth. It's a fairly mild model of stellate cell activation, but nonetheless, we get significant differences. Then when we do the opposite, we inhibit stellate cells. Here we knocked out YAP. YAP is important for fibroblast activation, but for stellate cells only in some models, not in all of them. So when we knocked out YAP, we see again less fibrosis and less stellate cell activation. the stellate cell activation here, a DN and CCL4 model, so this is a fibrotic model, we see less tumors. Then we wanted to also not only have the DN models. My lab is moving away a little bit from DN models only. This is a spontaneous model, the MDR2 knockout. Here we needed to knock out PDGF receptor beta. We tried also the YAP, it didn't work. When we knock out PDGF receptor beta, we have less fibrosis, probably because stellate cells proliferate less, not because they activate a lot less. So we have less stellate cells in these livers and we get less cancer. Then the third model is the depletion I just showed to you. We wipe out the stellate cells, which works fairly efficiently, a little less in the chronic than in the acute setting. We wipe out stellate cells, we get less cancer. And since we didn't want to use the ALRED all the time, we used another mouse, alpha SMA TK mice. These are from Raghu Kalluri's lab, who had used them in pancreatic cancer. These kill the proliferating cells, the thymidine kinase, in this case, the proliferating myofibroblasts that express alpha SMA. We can wipe them out quite similarly, actually, to the ALRED-Cre, not a big difference. And we also see that we have less tumors growing. So basically, this took only like 5 years or so, but I think all the data kind of show the same thing, that stellate cells promote cancer, and most likely it's the activated stellate cells here, because that's what we really change mostly. So, but the question is, how do they do that? And so we did some single-cell RNA-seq to better understand that. I mean, basically, we didn't reinvent the cells. These are quiescent and activated stellate cells, which we call my and psi HSCs. And it's a gradient from more activated to less activated, which we saw in multiple models and in patients. And why did we call them psi HSCs and my HSCs? Well, this is just, based on a single, oops, I'm trying to go back. It does work, yeah. This is a single-cell RNA-seq data. So the MyHSCs, obviously myofibroblastic, express ECM genes and also some contractile genes. But these more quiescent genes, we were surprised that they have a high amount of cytokines, growth factors, and inflammatory mediators. That's why we call them PSI-HSCs. So it's really just a functional nomenclature describing well-established cell states. And not surprisingly, the quiescent PSI-HSCs, they go down with progressive liver disease here in mice. We've also shown this in patients. And the activated MyHSCs, they go up. So that's expected. And then we try to better understand the mediators in these cells and try to understand whether maybe there are mediators from quiescent or from activated cells that can influence this. And we did a lot of different approaches, and there's not only collagen and HEF. We knocked out several of the candidates, but these are the ones where we got strong data. So collagen 1A1, as I said, in the activated cells, and the HEF in the more quiescent cells, and that's true for all the models, and is kind of, a gradient, but where we have more collagen, we have less HEF and vice versa. So they're kind of inversely correlated. And the same is true for patients. And then when we look at the activation, HEF goes down with activation, collagen 1A1 goes up with activation. So the question was then, we established this data, but what do they actually do? So we first focused on the type 1 collagen in the activated cells. And here I'm showing you again the MDR2 knockout model. So the mice at about a year or 14 months, they start getting tumors. And so we knocked out type 1 collagen, which is quite efficient. In the beginning, we used mainly the MX-Cre mouse model, which knocks it out in the entire liver and even some other cells in the body. We have less fibrosis. And so then we get less tumor development. This wasn't significant in all parameters, but the tumor number was significantly down. We did a bunch of other models. So the usual DN-CCL4 model, and then we switched more to the ALRED-Cre. And so here we see, again, strongly reduced fibrosis and a clear difference in multiple parameters, so basically all parameters that indicate the liver tumor burden. And the strongest data we saw when we deleted collagen in this NASH model with the CDAA high-fat diet, where we see really highly significant differences in the tumor number and tumor size. In a fatty liver model, you don't usually see a change in the liver body weight ratio because most of the mass comes from the fat. So overall, we see in multiple models that collagen has a role in promoting liver tumors. And again, how is that in patients? Well, fibrosis is actually associated positively with increased risk for HCC development. So that kind of made sense, but it had never been functionally proven. So the question is then, where does this happen? Let me just go back. So we have fibrosis in the tumors. We have fibrosis outside the tumors. I'm not sure. I think I don't have the slide here, but most of the fibrosis in HCC is outside the tumors. So, but we looked at this, and what we found is that collagen 1A1 deletion reduces stiffness. So when people in the mice sacked the people in the lab sacked the mice, the livers were always softer, and we measured this formally. We found that specific mechanosensitive signaling pathways were altered, especially YAP and TAZ, but by the immunohistochemistry was more TAZ than YAP. I think YAP is a lot in the cholangiocytes. TAZ is more dynamically regulated in the hepatocytes. And we knocked out TAZ and YAP as key downstream mediators in the hepatocytes. I'm showing you only the TAZ data, which was positive here. So when we knock out TAZ, we could reduce HCC and proliferation in the non-tumor environment. So basically, just to explain to you, the livers are usually very stiff. That affects the hepatocytes. They like to proliferate, and that somehow promotes tumor formation. When we knock out collagen, the livers get softer. We have less TAZ, less proliferation. That's kind of what happens. So then the other question was, oops, the slide we already have, there's some duplication. I think I'm going a little bit back and forth, but so the other mediator was the HGF, which we knocked out also in the stellate cells. And since HGF is a growth factor, people have probably heard that MET, the receptor for HGF, has been a target in HCC. So our expectation was when we knock out HGF, and Sylvia actually had shown that in the cholangiocarcinoma, we would get less tumors. But that wasn't the case here. So we knock out HGF, that works quite nicely, but we get this drastic increase in tumors. So what's happening here, so this is really probably some of the most significant changes we've ever seen. What happens is that we have more injury. So we have much higher levels of ALT in this DN-CCL4 model, or just CCL4. And we have more tunnel positive cells. And so the cells, when we don't have HGF, the hepatocytes die more. We probably get then in response more proliferation, also more inflammation, we get more tumors. And we also tested this, and this has been published many times before. When we add HGF to hepatocytes, it's very, very protective. So together with our regeneration data, we really think that the main function of HGF in the normal liver is not the proliferation, but the protection from injury. So now I'm getting back, sorry, slight mix-up in the slides. Back to the collagen. So I showed you before that we saw some differences in the non-tumor tissue. That actually was a slide I already showed you. Not sure what happened here. But we also looked at additional mechanisms. So one is the stiffness for collagen, but collagen is also a signaling molecule. So collagen can activate different receptors. We looked at a couple of receptors, integrins and DDRs. I'm only showing you the data for DDR1, which is a collagen receptor. And basically, we knocked out DDR1 in the hepatocytes using an AAV-CRE. So that's hepatocyte specific, and then did our model. So we didn't manipulate the collagen, but the collagen receptor in this case. So DDR1 deletion did not change the fibrosis. The depletion was quite efficient in the hepatocytes. Fibrosis remained the same. But what we saw is a difference in the in the tumor development. So DDR1 knocked out in the hepatocytes leads to a strong reduction in tumor development, which is kind of similar to the collagen deletion, maybe a little bit less. When you look at patients, and the TCGA data set is not perfect, and maybe there are gender differences, and in the mice we usually use male mice. So when we look at overall patients, there's kind of a strong trend, but when we look only at the male patients, we also see that high expression of DDR1 is associated with worse survival in patients. Then we wanted to look a little bit more at the mechanism, and here it's exactly the opposite. So when we look at the non-tumor cells or non-tumor environment, we don't see differences in the proliferation here. But when we look at the tumor itself, we see differences in proliferation. So DDR1 knockout has less proliferation. And DDR1 is also more highly expressed in the tumor cells. Didn't show this, but it's in the paper. And then finally, this is together with Michael Karen. What we found is that probably DDR1 is regulated not by just collagen, but by collagen degradation products. So what he found, and also found in our setting in the HCC, is that when we have ECM on in the environment, he saw that there's an upregulation of DDR1 activation. And so the cells plated on ECM versus plastic. But when he plated cells on a non-degradable collagen, this is this RR variant, DDR1 is lower, phospho-DDR1 is gone. So this non-degradable, non-cleavable collagen cannot activate this receptor. And then we looked at the different collagen-degrading enzymes, MMPs, and we saw that they're usually highly upregulated in the tumor, often much higher in the tumor than in the non-tumor parts. And looking also at patients, we could see that the patients who have a high level of these collagen-degrading MMPs in the signature, they have a worse survival. Obviously, MMPs also do a few other things, but we think this correlated quite nicely. So to summarize this, we think that collagen has two roles in the outside tumor or pre-tumor environment. Collagen creates a high degree of stiffness, activates TAS and promotes proliferation. And then in the tumor itself, it activates a receptor, DDR1, that also promotes proliferation, probably also some resistance to cell death. And this is, we think, mainly due to stimulation by collagen degradation products. So who wins in the end? Coming back to the kind of bad and good guys, I think it's a philosophical question because we don't know. It's possible that actually a lot of the patients who have cirrhosis and never develop HCC, that protective mechanisms are in place and make things, happen a lot less. But what we know is that when we have low HEF levels, we get more tumor development or faster growth also. And especially when we looked at two data sets, these are bulk RNA-seq data, so it's not perfect. But in HCV and back then still NAFLD cohorts, we see that there is a disbalance often. And when the disbalance is high, we have a higher risk for HCC development. So when we have more MyHCC, less high HCCs, so basically more bad guys, less good guys, risk for cancer development is higher. So to kind of summarize the data, I also have a slide with a cholangiocarcinoma. I didn't put that in, but I can explain it a little bit. So we have different types of fibroblasts in the liver, the MyHCCs, the SciHCCs, and then we also have mesothelial and portal fibroblasts. But we only focus on the stellate cells. So these MyHCCs that are activated produce collagen, and through two different pathways, one stiffness pathway, kind of pre-malignant environment, and one DDR1 receptor pathway in the tumors promote either tumor development or tumor progression. Data I didn't show you. There are other ECM mediators like hyaluronic acid, But in the HCC, that was pretty weak. We had to do a lot of mice to see a minor effect. In the cholangiocarcinoma, this was much stronger. And then there are these quiescent or more quiescent Cy-HSCs that produce higher levels of HGF, and they protect the hepatocytes and they protect from tumor development. And I just showed you the R-spondin-3, and there are probably other things that we need to study more. So I think we kind of have multiple mediators probably in the activated cells, multiple mediators in the quiescent cells, and it's pretty complicated. So we need to go one by one and do all the different studies. In the cholangiocarcinoma, clearly some of these functions are different. Here, HGF is protective in the cholangiocarcinoma, and also in liver metastasis, HGF was disease promoting. And for the collagen in cholangiocarcinoma, we didn't see a clear effect, but in metastasis, we saw that it was the opposite. It was restraining the tumors. So it's really very much context dependent. The cells are quite diverse, so we can't really say stellate cells promote tumors. There's, I think, in every tumor a balance of these different states, but then in the different tumors there are also differences between them. So that was my last slide. 10 minutes time for questions. I still want to mention everyone in the lab who has done the work. So Yoshi and Atsushi are the ones who really did the main work on the liver zonation and liver function with the stellate cells, but this was actually started by Juan in the lab. And then Aveline was the person who did most of the work for the HCC. Also want to thank all our different collaborators. As you see, a lot of people from Europe here and our funders, and I'm happy to take questions. Yeah, so, I mean, we never saw anything black and white, and I think we tried some single cell analysis and see can they go back and forth. And I don't trust this kind of analysis 100%, but our opinion is yes, the cells can go back and forth, and that one of the main regulators also described for other cancers is TGF-beta, as you probably would have suspected. That's the main candidate. We currently do not have a good way of knocking out TGF-beta in the stellate cells. I mean, we have actually done it in vivo, but because our CRE also has activity in some other places or for other reasons, these mice over time develop quite a few abnormalities, so we chose not to include that. But I do think that TGF-beta is probably the main, but not the only regulator. So we're working on better ways and addressing that, yes. And so I think it's a very plastic system going back and forth. Yeah. So we have a little bit of problem finding the senescence in the mice. I can't talk about patients. So actually one of our reviewers for the paper said, well, show the senescence. And we did a lot of staining. We found a lot of senescent cells in the liver and the mouse liver. But a lot of those were hepatocytes or macrophages. The stellate cells didn't show that so much. I still think it makes sense because stellate cells, fibroblasts, they proliferate a lot, and I think a lot of in vitro senescence has been shown on them. So I can't answer that very strongly, but what I think is if senescence occurs, I don't think they would go back, have a SASP phenotype with high HGF. I would think no. I would think that this is probably a third phenotype in the single-cell RNA-seq, and they'd probably have a lot of inflammatory mediators but low HGF. But that's speculation. The first part, I don't know if you saw that slide. I don't have data for the HCC in here now, but this is a nuke-seq. These are also samples that you provided from Pittsburgh included, but multiple things. So there's a mix of different livers, and when we look at least this setting, mostly end-stage liver disease, we do not see a lot of gene expression in other cell types in terms of collagen. So, and this is not showing the collagen, so we still need to, we have it, but I don't have it here. But we think that the stellate cells, at least in end-stage liver disease in patients, are the main fibrogenic cell type. I don't think it's the hepatocytes. I think the cholangiocytes can maybe make a little bit, but in some, disease states that can be different. In the cancer, it's still true. We've done that mainly in the mice, but cancer cells can make a little bit of collagen. And there's this guy, Raghu Kalluri, who has actually published a kind of strange paper. Sylvia probably likes it. They knocked out collagen in tumor cells. And they say tumor cells make a different type of collagen. And usually collagen is the heterotrimer of the type 1 collagen, of the collagen 1A1 and the collagen 1A2 chain. And they say that the tumors make only homotrimer, so collagen 1A1 homotrimers, and that these are actually very different biologic activity. Not sure if that's true, but apparently the tumor cells can make it. That was in pancreatic cancer. So yeah, we have to, I think, analyze a little bit more the tumor setting to do that, or the very advanced diseases, and focus on epithelial cells, but I think they're always going to be like making very little. Yeah. Second question with the checkpoint inhibition. I think that's a really interesting question. I didn't include this data here that in the mice, we have very little fibrosis, very little stellate cells in the HCC. There's some peritumoral fibrosis, so that alone already could regulate the immune cells. In the patients, we have more, like always, we have a bigger range. So overall, outside the tumor, we have more fibroblasts and fibrosis than inside the tumor. But there are people who have fibrotic tumors with quite a few fibroblasts and ones that have less. And so I think there's a good chance that there is a regulation. In our mouse studies, we didn't see so many differences in immune cells. We saw a few, we're following up. But I know that in other tumors now, people, especially the pancreas, people have inhibited fibroblasts and see quite drastic differences with the checkpoint inhibitor responses. So I think it's worth following up. I think in tumors, like just targeting, at least in HCC, targeting the fibroblasts probably is not going to make so much difference. But I think maybe the combination therapy could be something. Yeah. All the cells in the liver talk a lot to each other. That's normal. Like we talk a lot to each other. We don't only talk to one person. But it's actually well established that, anatomically, stellate cells and endothelial cells are very close together. And there are a lot of studies showing that as a functional unit. And I think especially for the Wnt pathway that I mentioned, and that has been shown, Wnt are highly, and deleting Wnt and stellate cells didn't do anything. But Wnt are highly expressed in the endothelial cells. So I think for this specific pathway, the R-spondin 3 comes from the hepatocytes, from the stellate cells. The Wnt come from the endothelial cells, and the three together form like this network that then regulates the zonation genes in the hepatocytes. So it's not stellate cells alone, but I focused on the stellate cells because their role in regulating hepatocytes was not known. Yeah, I can only speculate. From your work, Sylvia, we know, and from others, that these tumors are highly desmoplastic in the tumor setting. We also know that many cholestatic liver disease have a lot of fibrosis. But we never figured out what are the signals, just because it's technically more difficult. I still think it's mainly TGF-beta, but there are 3 isoforms of TGF-beta, so that's hard to knock out. But we honestly do not know. It could be something else. So I think you should ask Sylvia that question in 1 year, and she'll answer it.
Perfect. Thank you very much to everyone.